introduce Anne O'Donnell-Luria, who is giving our next talk. And I think, Anne, if you just want to come over, oh, look, we're doing a mic transfer right now. So Anne is an attending physician in the Division of Genetics and Genomics at Children's Hospital. She's an instructor at Harvard Med School and associate director of the Broad Center for Mendelian Genomics. Her talk today is Annotation Data and Its Uses, and she will also be leading the filtration workshop after the break this morning. So with that, I'll turn it over to Anne when she is all set.
Thank you, Danielle. Okay, so this is a very much whirlwind talk that is going to take us through many types of annotation data, which I will go into in varying levels of detail. But a lot of this is to try to start from the beginning and a lot of the major concepts, and then give you a hint of all the things and the annotations we'll be using in the filtration workshop that comes after this. So I have no disclosures. So we'll be talking about what annotation data is, the issue of transcripts, which are very important biologically, but in terms of interpretation, make things more challenging. We're going to be talking about genomic reference databases, mostly Nomad, and the various features that we use in that. A little bit about in silico predictions, and then I'll be introducing a bunch of databases at the end very briefly to give you a hint of things. So when we do exome sequencing, like Daniel told you about, the output of that is a variant call file, which is just a list of all the places that the sequence differs from the reference sequence, which is the reference human genome sequence that has been determined as the standard. There are many different types of variants that we can find. These variants will fall in different transcripts, and then they'll have different frequencies in the general population, and we can predict they have different consequences based on in silico predictions. The types of variants, just to give you an example of what I mean by types of variants, so the reference is listed at the top, a missense variant. Hopefully everyone in this audience knows this, but I just wanted to start at the basics. A missense variant is something that changes the DNA and the RNA and the amino acid. You'll see I messed up this slide. The synonymous variant should also change the DNA and RNA, but does not change the protein. And then a frame shift inserts a base into the DNA and RNA, which causes everything downstream to change in terms of the protein. And a nonsense variant changes an amino acid, a base that causes a stop codon and truncates the protein. So we use these two types of nomenclatures, VCF nomenclatures, but the other is the Human Genome Variation Society, or HGVS nomenclature. And that's the more common one clinically that we'll see. It's the C dot that explains the DNA change, and the P dot, which is the amino acid change. And there's a lot of rules that we follow with these nomenclatures. The HGVS and the VCF don't really perfectly match up like you kind of might think they would. So HGVS annotates versus the transcript. So it can, and it's very important to know what transcript you're referring to. The VCF annotates based just on the genomic coordinates and always refers to the plus strand or the top strand of DNA, the 5' strand. For HGVS, we have silent variants that we just represent mostly as the P dot. And then I like these ones where we have the actual position and that it doesn't change. A missense variant, isoleucine to valine here, and it gives you the codon position. And then for an indel, you can see where it describes for the C dot the bases that are deleted and then the actual change for the protein. And just to mention, in-frame indels are when you delete three bases. So you delete one amino acid, but you maintain the frame of the protein. And in general, I kind of think of these like missense variants because it's just sort of changing one amino acid. We also have nonsense variants, which we'll see represented these three different ways. That all means that there's a stop codon that's getting introduced. A frame shift is written these ways with the frame shift. It's nice to kind of, when they have how many bases after the frame until you hit a stop codon. And then splice site variants are written as the nearest C dot position, and then whether you're minus or plus. And we'll go into that a little bit more in a few slides. So HGVS, again, refers to the transcript and aligns everything to the right. And a VCF actually aligns everything to the left, which is how you can get these sort of discrepancies between the two systems. So in a VCF nomenclature, so this is a CFTR, Phe508del, so it's the most common variant for for cystic fibrosis, and we'll go into this variant more actually in a little bit. But just to show you here, so in a VCF, we kind of anchor to the left on this A, which is not deleted, and then we delete this TCT. And so it's written like this. But in HGVS, it's actually, we anchor to the right. And because what's left at the end is these two T's, we actually can't tell sort of which T's got deleted, and so we just anchor to the right for that. And so it gets reported the CTT gets deleted. So why does this matter? The biggest thing is if you're like looking in a database and you're using HGVS and the database is using a VCF nomenclature, you'll think that you're not looking at the same variants. And so it's really important that you're annotating your data in a way that it is aligning with all other data, so you're getting the right annotation on each variant. Another complication is genome builds. So we have a consensus human reference sequence. There's large groups that work on improving this and filling it in. For the most part, genes aren't changing at this point, but the space between genes is still getting filled in with additional detail. Additionally, haplotypes are kind of, so regions that are very variable between different populations are getting filled in also, and so the genomes are getting more complex. Just to show you an example of what this looks like, this is that same variant we had on the last slide. HG37 is the build that a lot of us are still working in. HG38 is the kind of currently recommended genome build that we're moving towards. And you'll see that these are, I mean, the actual base changes are the same, but the positions are different, and these are actually 350 kb apart. So if you were to look at a variant and look in the wrong genome build, it would like typically appear in like a totally different gene than you thought you were looking in. And so it's very important to know what genome build you're working in. And then this just happens to be the case because where we've been moving all our data over to HG38. So our genomes are over, and the genomes I'm getting to analyze in the filtration workshop are in HG38, and the exomes are still in HG37. And that's kind of the real world that we're living in as we move everything over to HG38. So there's a number of quality metrics that we know about variants that we have in our VCF. This is an example of how it looks in our system, which is called Seeker. So we have like a ID for the sample. A is the reference. It's not in bold. The bold is the G, so that's the alternate variant. And this A/G means the patient is heterozygous for a G at this position. If they were homozygous, it would say G/G. They'd both be in bold. And if they were homozygous reference, then it would say A/A. So one of the first things we have is the genotype quality. Ours are written down here. 99 is the maximum, so that means it's very high quality. We generally look at variants that are greater than 20, but honestly, like you can have real variants that are lower, and you can have artifacts that have higher GQs, but it's an important metric to look at. Depth is the number of reads at the site. You'll see that here, so there's 54 reads over this site. The allele balance is another important feature. It's the number of reads over the site that have the alternate variant over the total number of reads over that site. So this one right here is 31% of the reads have the alternate variant. So it's sort of within the range. So for something that's heterozygous germline, you'd expect about 50%. But there's always like a distribution there. And so 31% isn't hugely out of line, but it does raise a question of could this variant be somatic in this patient? As you go even lower, your chances of being somatic increase. And then VQSR, it's not on here. Generally, things that have good VQSR scores, it's not written down, but you'll sometimes see when you look at the data, they'll actually list a VQSR score. That's when it's failing this filter. This is basically a quality filter that asks the questions, the variant look more like a good variant, a real variant, or more like a bad variant or an artifact. There's more information about this if you want, and all these links are available. The slides are available, so you'll have these links. But just ballpark, that's what it does. So transcripts are incredibly important. and variant annotations may be different like a variant doesn't necessarily fall in every transcript for a specific gene, and it can have different impact. So this is just an example here. This is MECP2. The other thing I wanted to mention here is we have two main sets of transcripts. One is RefSeq, that is which is sort of American NCBI based, and the other is Ensemble, which is GenCode and more from European annotations. RefSeq tends to have fewer transcripts. GenCode tends to have more. We use GenCode in some of the databases like Nomad that I'm going to talk about. Generally, the canonical transcript is kind of the transcript we all talk about and refer to as sort of the main transcript, but it's actually just typically the biggest transcript, and the one that includes all the exons, so you're kind of referring to everything. It doesn't necessarily mean that that is the clinically relevant transcript, so it's just important to remember. There are groups that are working on defining the clinically most relevant transcripts, and that's going to be very helpful when that's all available. That still won't mean that for your specific disease that the most clinically relevant one will actually be the most relevant for your disease, but we're making progress in that area. Okay, these are some of the in silico predictors that we use in the seeker system that you'll be playing with in the next session. They overall, a lot of these use the same training data, a lot of similar metrics in the models, and so there is a lot of circularity to using multiple in silico predictors. On the other hand, it's sort of better to have more data than less data. That's definitely the Broad approach to things. And so we tend to look at a lot of in silico predictors, but we don't think of them additively at all. We just kind of think a little bit about consensus, and then there's some we trust a little bit more than others. But for a specific effect of a variant, it's really hard to know which one you should care about the most. I'm not going to go into the details of any of them. I will say some of them are only for missense variants. A few of them come up on all variants, and so that's helpful just to have a score for each variant. And then there's some new Splice AI scores that are actually developed by Illumina that are supposed to predict variants that are cryptic splice disrupting variants. The other thing we always look at, which goes into a lot of these scores, but we still like to look at, is conservation. So going over to the UCSC genome browser and looking at your amino acid, if there's a missense variant there, looking to see across many species what we see there. Well, the tools will use conservation. The reason it's kind of helpful to look is, so this is a serine in almost every species, and if I had, say, a histidine there, I might say, oh, that's a very different change. But if I had a cysteine, then, well, I do see that in 2 species, and so maybe that specific change is more tolerated there. And so it's just helpful to know. For variants that alter splicing, we tend to think of, so this is an exon, the intron, and then the next exon. So this is the donor site over here, and this is the consensus sequence of it. So this GT here is very important, but also the 5th position tends to be more likely to be deleterious if that position is disrupted, and it's more likely to impact splicing. The splice acceptor sites, so at this side, which we call -1 and -2, are the A and the G, and those are also very important for splicing. There are other variants that it can impact splicing in the introns, also within the exons. Those are harder to spot. There are programs that do that, which I think is on my next slide. But the other thing I want to mention is just because you have something that disrupts splicing, you need to keep an eye out for rescue variants. We definitely have examples where, say this GT, or actually say this AG is deleted, but actually maybe it's just replaced by another AG. So it doesn't actually change anything, but in your VCF it would tell you an AG is deleted, there's a splice site variant, but if the resulting variant that's there instead is still an AG, it would be expected to behave the same. So it is important to look at the sequence context of what's actually happening. Aside from the in silico tools, we use Alamut, which is a software that is available for purchase through a license to be able to see variants that impact splicing. It takes 5 different splice scores, and you can put any variant in and see. So this is a donor site here, this AG that causes splicing right after it. But when we change this variant here to a G, now there's an AG there, and so it actually is, the predictors say that this is actually going to start using this AG instead of this AG as the new splicing position, in which case it's going to cause an in-frame an out-of-frame insertion of 2 bases, which is going to result in a frame shift. So these can be very helpful. These are all tools that are often publicly available, but we like Alamo because it puts it all together in one place and nicely visualizes it. Okay, so Daniel's talked to you a lot about exome sequencing, but basically when we sequence the 20,000 human genes, we end up with over 20 to 30,000 protein coding variants that are high quality, passing QC filters. And there are many, many rare, potentially functional variants in every person's exome, including all of us in this room. We all have about 500 rare missense variants, and when we use these in silico predictors that I showed you before with the red and green dots, about a third of these are predicted to be damaging by in silico predictors, so we obviously we don't want to consider those too heavily in the weighting. We all have about 100 loss of function variants, actually about 20 of which are homozygous, but that does count things like olfactory receptors, and we all have about 20 rare loss of function variants. We have 100 rare variants in known disease genes, so if you're thinking about looking at a large panel of genes associated with a phenotype, by chance you're going to have a rare variant in some of those genes, because we only know about 3,600 disease genes so far, so 1 in 36 of those is going to have a rare variant. And then we have 50 reported disease-causing mutations, which is an astounding number, and I don't know how you all got out of bed today and made it to this course with all of these mutations in you. But what basically is happening is a lot of the databases that we really rely on have been filled with a lot of variants that were found early on before we had good reference population databases. And so people from ancestries that didn't have good control populations would go to clinic, get sequencing, they would find a rare variant, but it would really turn out to be a variant that's very common in, say, East Asians, and we didn't have those controls before. So some of that is getting cleaned up, but is still an issue. We all have 1 to 2 de novo coding mutations. These are obviously a place we look more carefully for variants that could be causing disease, but it's important to remember that we all have these, and so just because there's a de novo variant doesn't mean it's causing disease. And then we all have an unknown number of sequencing errors. So how in the world do we find the pathogenic genetic variants that are causing disease in our patients in this sea of so much benign variation? And we'd all like a tool like this where we just look at the data and we can just see where the pathogenic variants are. And occasionally that happens when something's in a clinical database and everyone agrees that it's pathogenic. But a lot of our current approach is sifting through a lot of data while getting a little cross-eyed. And we have tools, and there are many tools in the field that are helping to make this all easier, but it's still a process that requires a lot of human input and thought. Okay. So one of the biggest things that Daniel's lab has led and has been really powerful across the field is comparing one patient or one family's exome data to tens of thousands of exome data, so we can figure out which variants are really very rare and which ones are more common. So there's a number of reference population databases that have been available. I'm just going to briefly go through these, particularly the ones that we use still in our rare disease analysis. So 1,000 Genomes is the very first reference population database. It's, it was when these sequencing technologies were just becoming available. It's exomes and low coverage genomes, but they're from really diverse ancestry, so it captures a lot of genetic diversity. EXAC was the first aggregated exome database. It has 5 different ancestries. Again, it's from the Broad Institute and led by Daniel MacArthur. And it really changed the field and became the standard reference database for molecular diagnostic labs and researchers. I will say ESP was another early sequencing effort. We actually don't use this in our annotation because much of the ESP data is in EXAC, and so we wouldn't want to double count. Bravo is a whole genome sequencing project from the TopMed, from NHLBI. The data is available through a website. It is HG38 native. It's the only currently available HG38 native data set, and so it's been really helpful, and you'll see it annotated in the data you'll be analyzing next. There are a few related, they haven't pruned their data for related individuals, so that can inflate rare variant frequencies. We're not sure how big of a problem it is. And then they have some restrictions on data sharing, so you can't actually see the ancestry of the individuals who have a variant. And then Discovery EHR is a different type of data set. It's from the Geisinger Regeneron, and it's connected with electronic health medical records, so you can do interesting studies of phenotypes along with variants, but a large proportion, and potentially the majority of their cohort actually is related because they're all from kind of the same geographic area, and so you have a lot of relatedness in there, so you would not want to use that for a filtration for your population frequencies. And then NOMAD is really what we use today. It has over 140,000 individuals, 125,000 exomes, and 15,000 genomes. It's data that's been sequenced in other projects, but shared for this aggregate use for the community by 109 different PIs. The Broad Institute processes all the data through the same pipeline and jointly calls all these samples for an aggregated release. This is a huge resource that a huge amount of people work on and getting this to the community, and so I just want to highlight them here. Particularly Conrad, Laurent, and Grace spent a long time QCing this data so that we could all have it to use, and then Matt and Nick are the ones who put it on the browser, which is the main way I access the data as a clinical geneticist, and then Jessica is the person who gets us permission to use all this data, which is a huge job when there's 140,000 samples. So this is the principal component analysis from Laurent, which basically looks at the variance between the samples. The largest driver of variance, genetic variation, is ancestry. And so here we're able to differentiate the different populations, and we determine ancestry from the genetic data. It's not self-reported in this. And so you'll see over here, we have the European samples in orange with the Finnish and Ashkenazi Jewish bottlenecked populations nearby. This is African American down here. South Asians are up here. Those East Asians over here, and I think Latino. And so we're able to pull out all these ancestries this way. We also remove low quality samples from this data set and any samples with sex chromosome abnormalities and first and second degree relatives, again by genetic data. So these aren't completely healthy people. I don't know where we would find 10 completely healthy people, let alone 140,000. So these are people who were sequenced in other types of studies, so type 2 diabetes, people who had heart attacks, people who have migraines, there are schizophrenia, people with schizophrenia samples in here, but they should be depleted for severe pediatric onset disease. So we have not put any cohorts in here that were recruited for pediatric onset disease, which doesn't mean that you couldn't have some type of, pediatric vision impairment that's monogenic and be in a type 2 diabetes study, but you shouldn't be enriched for any of that. So you should be more similar to what you see in the general population. So we think pediatric disease should be depleted in the data set, but not entirely removed. We don't have individual level data that we can share, and we don't have phenotype data. The amount of data we do have, we've put a lot of it onto the browser, and so I'll show you that as we go through. The male-female is pretty balanced. It's about 55% male samples. The mean age is 54 years. We also use something called PopMax, which is we report the allele frequency in a sort of single line of the highest of the main continental populations, the highest allele frequency, because we think of something as very common in Latinos, but rare in other populations. We would still think that would be unlikely to be a cause of Mendelian disease, and so we would use the PopMax allele frequency. And you can download this entire data set. It's huge, so you don't want to just download it for running on your computer, but if you have an annotation pipeline, this is a great data set to download and annotate all of your exome data with, or genome data. So this is how I access the data for the most part through the NOMAD browser, which is just nomad.broadinstitute.org. And so I'm going to be taking you through how we use that. So this is if you type in the gene CFTR, this is what the browser looks like. Can I actually get a show of hands of how many in the audience have never used the NOMAD browser? Okay, great. Sorry, I just want to make sure at least it's worth going through for some people. So that's helpful, thank you. Okay, so what is on here is this is all the exons in the CFTR gene. And then these nice little mountains up here in blue are the exome coverage data. So it goes up to about 100x at the highest. And so you can see the exome data is, you've got these peaks. The values in between are introns, which we're not expecting to capture on the exons. But you can see the coverage is variable. And then the the green line here is the genome coverage, which you can see is really nice and smooth. Like genomes, we get this really great coverage. The next thing we have is the ClinVar variants. So these are not ClinVar variants that are in the Nomad data set necessarily. These are just all the ClinVar variants that are path and likely path, the loss of function in red and the missense in yellow or orange and the other things in gray. And then here's just sort of an overlay of all the variants actually in Nomad. And then the biggest thing is it's just a giant list of this is the VCF coordinates, so every chromosome number, the position, the reference, and then the alternate, that variant, what the consequence of it is, if it's found in exome or genome data or both. So these are all 5' UTR variants because I'm at the top of the gene here. The number of times we see it, which is the allele count. The allele number is the number of people that we have data for at that site, but it's actually the number of chromosomes. So for autosomes, for anything other than the sex chromosomes, it's that number divided by 2 is the number of people. The allele frequency is the allele count divided by the allele number, and then the number of homozygotes we see for any variant. So we have a search bar here. So Phe508, we're going to go back to that most common variant that causes cystic fibrosis. When I type in Phe508, you'll see I get a few missense variants at this site, and I get that deletion, single amino acid deletion that is the common variant. And so when I click over there, actually you can see where it falls in the protein, which is sometimes helpful when I want to kind of know what's going on with where a variant is. I like to use that. And then if I just clicked on the coordinates that was over in that bottom corner, it takes you to this variant page, which looks a little bit different than this. I've kind of clustered together the parts I want to focus on. So you'll have the allele counts in the exomes and genomes and some allele frequencies. We also have a statistical adjustment of the allele frequencies because Nomad is a sampling of the general population. It's not saying like absolutely the actual allele frequency is 0.007%. It's saying that's the sampling, and so this is with some confidence intervals on there, a more conservative estimate. We'll have the read data, which actually has been down a little bit, but we normally have the read data on there that shows you, so you can see if you think the variant is real. We have the actual age distribution of people with that variant, and you can pick if you want to just look at the homozygotes or heterozygotes. We don't have age data on everyone, so sometimes you'll see that table is empty because it just depends what the study was able to share with us. This is the heart of each variant page. It's the allele frequency table. So it breaks it down by all of the ancestries, the number of counts in each ancestry, and then the allele frequencies in each ancestry. So I like this. So I actually just, I wanted to know how good was this as a carrier frequency database, like how representative of the general population is this? So we know the European carrier frequency of this variant is 1 in 41, which we just know because like many people get carrier screening for this. And it does vary by ancestry, but in Europeans it's 1 in 41. For this specific variant, it's higher for all of cystic fibrosis. And so this is the number of Europeans who are genotyped at this site, which is this number divided by 2, times 1 in 41. So we expect to see 1,543 carriers in the general population of this size, and we see 1,598, so that's pretty good. So it seems to be working well. Another thing I want to point out is we have about 9,000 East Asians genotyped at this site, or who have data at this site, and none of them actually have this variant. So it just shows you again that you can have very different allele frequencies in different populations. You can also we have subcontinent, so we have some subpopulations, which what is in here depends on sort of the sample representation in Nomad, but you can click on any of these arrows and see what other populations we were able to comment on, which can be helpful if you have a specific ancestry group. But the other thing we really have here is the number of male and females, which occasionally you also are interested in depending on your phenotype. So I told you people with rare disease, pediatric onset rare disease are depleted but not removed from this data set. So I wanted to see how it worked for this variant. So based on Hardy-Weinberg equilibrium, p squared plus 2pq plus 2 squared equals 1. So we actually expect to see 9 homozygotes. I'm going to skip the math for you, but just tell you 9 homozygotes from this size population. And we see 1 homozygote, which I was surprised by. I actually thought we would see 0, but we do see 1. And so So as I said, they're depleted but not entirely removed. So what is going on with this one individual? And so these types of databases, particularly biobanks, which is not what Nomad is, but biobanks where we have genetic information paired with medical records, are going to be a really interesting way to find these individuals and study them more. It's going to be interesting biology here. Is this person not penetrant for cystic fibrosis, or are they and they just participated in another study? So this is all the data I have to really get at that right now, is this part here. So you can actually, on each Nomad page, click some subsets and look in some different subsets of the data. If you're working on cancer, you might want to look in the non-cancer subset. If you're working on intellectual disability, maybe you want to look in the non-neuro subset. If you are also looking at the TopMed database, we also have this, so you can make sure you're not double counting things. But one of the other things we have is if you are listed as a control in a study, again, it doesn't mean you don't have other diseases because you're recruited for one condition generally in a study. So when you select this, it downsamples because we only have 60,000 people listed as controls in the study in Nomad, so it's more an XX size data set now. But you see that this homozygous individual is still there. So whatever study they're participating in, they're a control. So it is what it is. It's just interesting. And then it's always really important to look at the read data. I mean, is it possible that this is just a sequence artifact and I'm getting all excited and there's nothing going on? But when you look at this, and actually the coverage on this one is really deep. I don't have it all here. The reads look very clean. You may notice that actually most of these are blue and pink, so this is whether you're sequencing the forward or reverse strand on the coloring I have up here. And actually, if you saw this pattern, you'd be concerned that there's a big bias that you're only sequencing one strand. I need to make a new slide, so you're going to have to just trust me on this, but if you look at all of the data, there's tons of pink reads at the bottom, and so this is just some kind of sorting issue and I need to remake this, sorry. But basically, yeah, this all looks totally real. Okay, so constraint data. So this is another really interesting thing that has been done with having this large population data set. So this is work that was started by Caitlin Samoha and has been continued by Konrad Karczewski under Mark Daly and Daniel MacArthur. So the idea here is that we have a gene, or we have two different genes, the blue gene and the orange gene there. And these genes are present across the population. And mutations are going to arise in these genes at random, basically, not based on what the genes do, but more just a little bit based on their sequence context. But basically, mutations will arise. And the difference you will actually see is that if you're a gene that is tolerant to variation, or most variants don't disrupt the function of the gene, then most of these variants will be passed on. But if a gene is constrained, where variants do disrupt the function and cause a phenotype, then most of that won't get passed on because of natural selection. And so when we look in today's population, tolerant genes will have a lot of variants, and intolerant genes or constrained genes will have very few variants. There's a whole complicated mathematical model to back all of that up that determines these numbers, but I'm going to skip ahead for the sake of time and tell you that on every Nomad page, we have this gene table that tells you for this gene, and I'm using NSD1 as the example here, which is a gene that causes Soto syndrome with its intellectual disability and overgrowth when you have haploinsufficiency or loss of function of one copy of this gene. And so what we have here is the number of synonymous variants expected, 525. We see basically exactly that, 523. So the observed over expected synonymous variants is 1 with the confidence interval here. For missense, we expect 1,400. We see 1,000. So we see about 75% of expected, which this is the z-score on that, so it is a little bit depleted. And then for loss of function, we expect 110. We see 5. So we see 5% of expected with the confidence interval here. And so this is very loss of function depleted gene, which makes sense because, as I said, loss of function of one copy of this gene causes this severe intellectual disability condition. Notes on constraint. This is for dominant disease genes. For recessive disease genes, we do expect to see loss of function variants in the general population. So you can see some depletion, but you won't see severe depletion generally. It's not necessarily a sign of a strong phenotype. It just has to have an effect on how often this variant is passed on. So something that just has a mild effect on fertility actually can have a very strong constraint score. So that's something to keep in mind. And then the way natural selection works is through reproduction. And so if you have a phenotype that is even very severe, but the onset is after typical reproductive age, you will not see an evolutionary signature for that. One of the other times that's come up is actually like, we have a cohort of vision loss. So in even sort of middle years onset vision loss, if it's not affecting fertility, people are still reproducing with this vision loss, then you're not going to see an evolutionary signature with that. Okay, so where am I going here? So this is going back to the browser. Oh, this is just showing you where at the top of each browser, at each gene page on the browser, we have this information. Other things we have on the browser. So we have the canonical transcripts here, with all the exons, but you can actually look at all the transcripts by just clicking the Show Transcripts box. And so when you do that, that shows you all the different potential transcripts. We're using GenCode, and we're actually using the last version of GenCode on HD37, and all of Nomad is on HD37, I should mention. And there is a leftover version of it that's available for download, and eventually the next version of Nomad will be on HD38, but right now we're still HD37 native. So here we have expression of each of these transcripts. So you can see one is very highly expressed and the others are more lowly expressed. And that is all from GTEx, which I'll talk a little bit more at the end. And then if you click, so this is the, this is the mean expression here. And if you click on that, you can actually pick, if you're interested in a specific region or tissue type, you can pick your area of interest. And then another thing you can do, so we have these drop-downs here. You have the drop-down, you can actually go to the original exact data. And there's one reason that I do this, and that is that the regional constraint. So we talked about missense constraint for the whole gene, but it's, loss of function variation, we tend to think, aside from things that happen right at the end that maybe wouldn't trigger nonsense-mediated decay, we tend to think of missense variants a little differently. And so if missense variants that fall in one domain or one region of the protein might be deleterious, and in another region they might be totally fine. And so Caitlin developed a metric where she looked at regional missense constraint. And so, for example, right here, this region here is only 4% of expected missense variation, while this region over here has 117% of expected. And so if you had a missense variant here, in a patient, you'd be much more interested in it. And so what this is showing here is that it's probably like a little bit hard to see, but these are all the ClinVar variants. And again, the protein truncating variants in red are throughout the whole protein, or throughout the whole gene, but the missense variants are really all piling up in this part here, and that's all within the very constrained, missense-constrained regions. And these regions are determined just by sort of scanning along the exons. There's no a priori knowledge of domains that goes into this. And we're working on updating that for the Nomad site, but Nomad data, but for right now, it's just the exact data it's available on. Okay, so then going back to the Nomad data. So I told you this gene is very constrained. We don't expect to see loss of function variants, and but we do actually see 5. And one of the things, if you notice the 5 up here, and that there seems to be, these are the loss of function variants here and where they fall in the gene, you'll notice there's more than 5. That's an important note, is the expectation model is really for essential splice site variants and nonsense variants. That we don't actually have an expectation model for frame shift variants, so we can't really tell you how often we expect to see a frame shift, because we don't really understand how, like, what's really driving indels appearing and where they should appear. So frame shifts are not part of the model, so this is only counting essential splice site and nonsense. So if you see these loss of function variants down here, you'll notice, so this is a forward strand gene. Another thing that's really important to point out here, sorry. So some are in the last exon, and maybe those don't cause disease because they generally nonsense-mediated decay is triggered by loss of function variants upstream of the last exon, because what triggers it is seeing exon-exon boundaries that still have the essential, like the exonic splice machinery still bound to it that don't get knocked off by the ribosome. This one here I looked at, and it's actually, there's a splice rescue right there, so I don't think this is a real loss of function. But then there's a bunch at the beginning here. One explanation is sometimes if there's a loss of function right at the beginning of a gene, you can have a rescue that, like, you can just start the gene from the 2nd or 3rd exon, and that might be good enough. That's actually not what I think is going on here. But I wanted to highlight Yeah, so I'm going to explain what I think is going on there, but I want to highlight one other feature first, which is that in VCFs, we annotate data. I told you whether we think it's a missense or a nonsense, but we actually annotate it versus its worst possible consequence. And so if you have an exon here that's only in one transcript and not in most of the transcripts, you might have a nonsense variant or even a missense variant there. And so you might see it, and it might be in a gene you think is really interesting. But when you really look into it, like, is that exon or is that transcript with that exon even expressed anywhere is really important to think about. And so we have a way that helps to do that. This is another example. This is being called as an exon. And so in Nomad, there's actually a bunch of splice sites that are getting called here. But this is, it's tiny. This is not a real exon. And so you have to actually look at the data and see if it makes sense. And you can see that this version is not expressed. OK, so this is work by Beryl Cummings, a former graduate student in the lab who's recently defended her PhD. And she basically wanted to say, can we take this transcript information, and she developed a score that estimates whether an exon is used in a transcript to help us with interpretation. And so basically what this does is it takes GTEX data for all the transcripts, and then it just adds it up for each exon. So if you have a variant here, she basically just looks across all the GTEX isoforms that are expressed across different tissues, and then she has basically an additive score for everything, coming up with a mean expression, and she gets one value per tissue. And this is so this data is now on the exec browser as this blue bar here. So anything that's sort of the full width of the blue bar is a highly expressed exon in most transcripts. This exon is not expressed. This exon is very lowly expressed or is expressed in a minority of tissues. And then this here, which looks really funky if you just look at it, what's happening here is that these exons are probably not really expressed, but these smaller things that are called exons within are expressed. And so these loss of function variants are all falling in that part that is between the real exons. So they're not actually transcribed and translated parts of the gene. And so they're not really, these would not be variants that would cause loss of function because they're not actually in exonic regions. And if you have a tissue of interest, you just click Show Tissues, and you can see if the pattern is different in your tissue of interest. For the most part, they've actually been pretty similar when we look. OK, so the other thing is we don't see ClinVar variants there, and that's why, because these are not real loss of function variants. OK, so Daniel mentioned the Nomad structural variant data set, which was released a month or two ago. And so I'm going to just briefly show you that. So there's a preprint out on bioRxiv. This is work from Mike Tolkowski's lab in collaboration with Daniel that was led by Ryan Collins, a graduate student, and Harrison Branda, an instructor in the group. So they did structural variant calling using multiple algorithms on about 14,000 genomes. It's a complicated method that I'm not going to go into the details. What I'm really just going to talk about is about 500,000 structural variants that they find in this data. It's about 8,000 structural variants per genome. About 25 if you take all of the loss of function variants, if you add them all up in Nomad and count the loss of function variants from SVs like deletions, the SVs make up about 25% of loss of function variants in the Nomad data set, so they're an important component. Most people don't have a large, so this is again a reference population, but actually 1 in 32 individuals has a greater than 1 megabase deletion in this data set. So there are rare large events that are happening, and so this data is all available to view. So this is what the SV browser looks like, and again, it's just at the top, you just flip over to the SV browser from the main site. So this is the gene RBM8A, which is a recessive disease gene for thrombocytopenia absent radius, or TAR syndrome. And most people with this condition have a common deletion that's seen in the general population in trans with a rare single nucleotide variant. And so I actually, I couldn't find good reference for how often do we see this in the general population. We know about it because we see it in patients, but we see it in enough patients that we know that this deletion is out there in the population. But how common? Does everyone have it, or is it only in 1 in a million people? How often do we see this? And so I went to this gene in the data set. So you can see, so this actually shows the gene with the intron-strontium scale. in this view. And so this is that deletion here, the common deletion. There's a list of all the deletions and duplications and everything here. You can color by type or consequence. So right now we're coloring loss of function are red. I can actually click and look at, actually I have to go into the variant page, but then I can click back and I can look at sort of this region. And so this is the whole deletion here, and this is the RBM8A gene here. So you can see it deletes that gene, but it deletes some surrounding genes also. And then I can see the coordinates of it down here. It's 240 kb, and we actually see it 3 times. And then we have a variant table, so it's seen in 2 Europeans and 1 African ancestry individual. So this is about 10,000 people here, so 21,000 chromosomes. So we see this in about 3 in 10,000 people, which is sort of consistent with what was estimated from the literature, but it's the first time we've really shown this in a large group. So we have this, and so for any time you're looking at a gene, you can come here and look and see what we know about it in the general population. Okay, I am definitely behind schedule, so I'm going to try to speed up even more. So frequency filtering is an approach that was designed by Nikki Whiffen, James Ware, and Eric Minikel. And it basically is the idea that when we use EXAC to filter rare variants, we can go from having hundreds and hundreds of rare variants to look through, that the bigger your data set, the fewer rare variants you need to look at. And the more you're able to say, I'm only interested in like super rare variants, the fewer variants you're going to need to look at, although it does kind of saturate out unless your data sets get bigger. So the question is really, how common is too common for a variant to be causing disease? And that question really matters, how common is your disease? Like for a disease that's seen more frequently in the population, you need to be thinking about higher allele frequencies than something that's very rare in the population. In the workshop, we'll often use about 1% for recessive conditions and 0.1% for dominant conditions. But if you know something about the disease architecture, you can go lower than that. So the frequency of a pathogenic variant in a reference sample should not exceed the prevalence of that condition in the general population. So if something occurs with a prevalence of 1 in 10,000 people, then you should be really thinking about variants that are less prevalent than 1 in 10,000 people, with some caveats. So penetrance makes a difference in founder populations specifically. This is the formula, which I'm going to skip through for time, but basically we have a calculator that James Ware's group developed that they share at cardiodb.org. It's the allele frequency app from that page. This is showing you for hypertrophic cardiomyopathy, which we know is a dominant or monoallelic gene. The prevalence of the condition is about 1 in 500 people. The most common variant that causes hypertrophic cardiomyopathy in cases, so how common it is among all cases, is about 3% of cardiomyopathy is due to 1 variant in MYBPC3. And so they're going to put 3% for their allelic heterogeneity, and they're going to put their penetrance of their condition at about 50%. And that will give them a maximum credible allele frequency of 6 times 10 to the minus 5, which is much lower than the 1 times 10 to the minus 3 that we generally use for our dominant filtering. So if you're really thinking about a specific disease area for those doing cohort studies, this is something that's worth investing some time into to figure out. Okay, so this is a list of some of the databases that we use, and I'm going to highlight a few of them briefly. So OMIM, or Online Mendelian Inheritance in Man, is a database that is a gene-level database. It has about 15 or 16,000 genes in there with information collected and human curated on a page. And additionally, it has every disease gene that's been reported for Mendelian phenotypes and some risk phenotypes. And so, NSD1 has been one of my favorite genes to talk about. And so this shows you the Soto syndrome. There's a page on that, including the clinical features, which are much, much longer. I've just highlighted a few of them here. And then this gene is also implicated in acute myeloid leukemia in the somatic mutation state. GTEx I've been talking about, so we use the data for a lot of things, but the other thing we can do is we can just go to the GTEx site and see where things are expressed. The other time I use this is sometimes if I have a variant in a gene and I'm like, I wonder if that impacts splicing. I wonder about doing RNA-seq. What tissues would be okay for me to do RNA-seq on? I can go to GTEx and I can look at some tissues. So in addition to sort of the like just human tissues from these autopsy studies. We have whole blood, and they also have some cell lines in here in the middle. So they have EBV transformed lymphocytes and transformed fibroblasts, so you can get a sense of which tissues might work out. And the other tissue we often use is muscle. Because you wouldn't want to query you wouldn't pay for RNA-seq to query a specific variant and then find out that it's actually very lowly expressed in that tissue. I don't know if I have this on a later slide, but in general, about 5,000 or 5 TPM is about what you need to really see impact on splicing, although the higher it is, the more likely you're going to capture splice junctions. The Monarch Initiative is a group that has put together a lot of model organism data into a database, and so there's tons of information on NSD1. We use this a lot more when we're looking at novel gene discovery, when we don't really know what the gene does and we want to get more information. So it collects data from a number of places. It has the papers or the database that the data came from and the information on the phenotype. So that can be a helpful place to look. ClinVar, I've mentioned, and you're going to hear a lot more about through the rest of the the rest of the course, is a place where clinical labs and research papers put their information in about how they're interpreting the pathogenicity of variants. HGMD, or Human Gene Mutation Database, is a pay access database, although there is a publicly accessible form that is basically has you don't have like the most recent things added, but you have everything that's like a year or 18 months old and older. And then LOVDs are databases that are often started by research groups where different people are putting information in. I kind of view these with like ClinVar, especially if there's a diagnostic lab and they've given their rationale for interpretation. I find that the most helpful. The Human Gene Mutation Database is literature curated, and a lot of this isn't using current standards from the American College of Medical Genetics for interpretation. And LOVD is often not curated at all. It's just more of a collection. There's another place you can go to, Genome2MP, which is Center for Mendelian Genomic Data. We don't have ours in there yet. We're in the process of putting it in. So right now it's from University of Washington, and that's led by Mike Bamshed and Jessica Chung and Debbie Nickerson. This is the site here. And basically, it's sort of kind of looks, it's got all your variants, and then it's got the number of HPO or phenotype profiles associated with each variant. So if I wanted to look up these 2 missense variants, then I would just click on them, and it actually tells me which HPO terms is associated with each variant, or if I had a variant that was seen in 2, and it tells me if it's in an affected or a relative. And so that can be helpful if you want to get information. And then you can actually contact the person who has the case with that variant and request more information if you think there's a match. So if I had the same missense variant or protein truncating variant in this gene and I was really interested in my patient had a nervous system phenotype, I might want to connect with them. And more data is going to be coming into data sets like this. So I think of it a little bit of like EXAC for rare disease as opposed to, so you don't want to filter on it, you want to use it more for matching. Whereas EXAC and OMAD you don't want to use for matching. Okay, so these are some of the many types of things I've talked about, and we use constraint, this data sharing like Genome2MP, functional data to push things in more towards being pathogenic. We largely use NOMAD with allele frequency populations and structural variant populations and the PEC scores to help us think of things, to identify the things that are benign, and then we have many, many variants of uncertain significance that we still need to spend time sorting out. So I talked a lot at the beginning about making sure your variant annotations and your VCF is all correct and everything's matching up and the differences between the different nomenclature sets. We use these reference population databases a lot in our interpretation, as you'll see in the next session. We all have many rare variants, so a variant being rare is not at all enough for it to be considered pathogenic, but it's a step in the right direction. And then I have shown you a handful of other databases that I think will be helpful that you'll get to dive into more in the next session. That's basically the workshop. Will go into this more. And then I have talked about the NOMAD team, but a lot of these resources and the workshop particularly was all put together by a huge team. Particularly wanted to thank Elena and Grace for putting a lot together and Danielle for all her work with this course. So I am happy to take any questions if I have a few minutes still. Thank you. Whirlwind of information.
I've used Nomad a lot, but I've never looked at the actual reads. Are these reads that you showed available for everybody to see, or? Yeah. Okay.
So in general, the read data is there. We don't have it for absolutely every variant. It's not, if something doesn't have read data on Nomad, it's actually just because the data was archived before we got to that step, and so we didn't have it in-house anymore. It doesn't mean that the read data is not reliable, but it is there for pretty much every variant. You can often look at up to 5 individuals with that variant too. And so I definitely encourage that.
And if a variant doesn't appear in the control tab, that means that it has some disease associated with it, probably.
It just means it was found in a person who was recruited for disease. But again, these are type 2 diabetes and migraine. I mean, they're things that are found in the general population. So, you know, the vast majority of variants found in an individual type 2 diabetes do not contribute to type 2 diabetes.
Okay.
Thanks for a nice presentation. I want to share one of the challenge we had with Nomad. Recently, we reported the first Mendelian form of non-alcoholic fatty liver disease. As you know, it's a multifactorial disorder, but we reported a dominant, autosomal dominant. We had 40 patients from 7 complex families. And we had a very nice segregation in mutation and phenotype. But when we checked the Nomad and exacts, we found for Nomad, Genomad, we found 170% with those pathogenic mutations. I mean,
170 people.
People.
Okay.
Yeah. 170.
Yeah.
With very definitely a stop codon and something. But the disease is a late onset. I mean, the full penetrance is after 40 years.
Right.
Yeah.
So there are I should like for cystic fibrosis variant the histogram, and it very much is like a well-balanced histogram with a peak at around age 55. So there are definitely younger people in the data set and older people. So you can look at the age distribution of your variant if you're interested, which didn't used to be available, but it's been available for a little while now. I don't know what you think the penetrance is, but also this is a phenotype that you would see in the general population. But it's also those phenotypes, it sounds like you have a lot of families, but those are the types of phenotypes where you really need like good cohorts for statistical analyses to compare prevalence of the variant in cases and controls. Great. So you're now all experts. I'm very excited to see you dig into analyzing the data.